Okay, then I think we make a start. Welcome everyone to today's biodiversity seminar given by Yann Hautier. It's my pleasure today to introduce Yann. He is an ecologist, mainly interested in the mechanisms by which global change affects biodiversity and how these changes then also affect ecosystem functioning and services. He's actually Swiss, so the first years of his career he spent in Switzerland in the western part. And then he did his PhD at the University of Zurich with Andy Hector. After that, he then moved to Minnesota and Oxford with a Marie Curie Fellowship. And then he became an assistant professor in Utrecht, where he's also now in the Netherlands. Yeah, Yann is very interested in these mechanisms and in ecological theory as well, and is involved in lots of different global collaborative research projects as well, like the Nutrient Network. He also founded the NPKD net. We might hear something about this as well. Yeah. And today he will talk about grassland biodiversity and ecosystem functioning in the Anthropocene, also presenting us some new results and new experiments that will now start, the BioCliVE experiment. And yeah, I'm really looking forward to your talk. And I would say the stage is yours. And yeah, welcome.
Thanks so much, Yann. Can you hear me? Yeah? Okay. So yeah, thank you so much for the invitation. I'm very happy to be here, to have the opportunity to present some of the work we are doing. So welcome. And today I would like to indeed present some of the results of my lab. And I would like to actually start by presenting some of the people that are working in my lab. Just make some move here. So of course, sorry. So we have John Connelly here and Bernard Schmidt here, so they are not part of my lab. But my students were very happy to meet with them at the GFO. So I just also put this picture here. So a lot of people are working together to make this presentation basically possible. So I would like to thank them already starting here. All right. So today what I would like to do is to show you the importance of biodiversity for the functioning of our ecosystems, and especially in the face of climate change. So I guess you know that we have a twin crisis, biodiversity loss and climate change. And so I would like here to put the state of what do we know about the importance of biodiversity for ecosystem functioning, but particularly around the stability of ecosystem functioning. All right, so I would like to start with a brief history of the relationship between biodiversity and ecosystem stability. And then I will go through some of the processes, how does it work, and trying to scale up from local to larger spatial scale. I will review some of the evidence and then also present some future challenges. All right, so the study of the relationship between biodiversity and ecosystem functioning is already quite an old story. Starting with Clement already in 1916, for example. And it has been very much a center of attention in ecological theory to understand whether biodiversity provides some stability in our ecosystems. And of course, it's also very much a societal and practical challenge because, of course, we can use this knowledge. If we understand the processes behind this relationship, then we can obviously use biodiversity as a safeguard against, for example, change in climate. All right. So the tricky thing with the relationship between biodiversity and ecosystem functioning, and especially the stability of ecosystem functioning, is that both biodiversity and stability are actually a multifaceted concept. So I guess you all know that biodiversity can take a lot of different shapes or metrics. You have biodiversity at the local scale that can be measured via species richness, Shannon index, Simpson index, etc. But also, of course, functional traits can play an important role in this relationship. Phylogenetic diversity, the community composition, okay, the similarity in species, for example, but also can take different aspects at different spatial scale. So that's what we're dealing here with, is all these different facets for just biodiversity. And the same thing apply for the stability. Stability can be measured at species level, can be measured at community level, at the meta-community level. So obviously, maybe different relationship appears to be at different scales that we look at. We have different definition of stability. Are we talking about temporal stability, which is usually measured as invariability? Are we talking about what I call the triple R, resistance, resilience, recovery, which are different aspects of this long-term stability that are usually measured over a shorter term? And of course, again, the spatial scale, which scale does this happen? So all of this has made the quest of the link between these two processes very difficult and quite challenging, but also with a lot of debate around that. So today, I would like to start focusing on local scale diversity, what I will sometimes call alpha diversity. And usually the metric that we use is species richness. And then I will also focus on temporal stability and the stability of biomass production. So you can see that we're already narrowing down quite a bit into these aspects. So I would like to make some definition of what we call temporal stability. So here I'm going to focus on temporal stability of biomass production. So you can see from this figure that here you have two species, the blue and the yellow species, and you can see that these species vary in time in their biomass production. So biomass production is measured every year at a certain time, and then it fluctuates as a response, for example, to environmental fluctuation, whether it's a dry year or wet year. And then, of course, these two components of the community would then make the biomass of the whole community. And then this biomass, of course, varies depending on this variation of the species level. So we can measure stability at these two scales, species level or community level. And the way we usually do that is that we use the coefficient of variation, okay? Which is basically taking the standard deviation, so that's measure of variability, and then we scale it by the mean. Normally, when you have a higher mean, a higher biomass production, you also have a higher variability. There's this thing going on. So we scale by the mean so that we can really have an idea about how much this is variable depending on the kind on the mean. And then if you take the inverse of this coefficient of variation, basically you have the temporal stability, which is also called invariability. Okay, but basically that's measured using the inverse of the coefficient of variation. So that's what that's the metric I'm going to use when I talk about temporal stability. Now, this is the first evidence from experiment linking biodiversity, species richness here, and the coefficient of variation, so the variability, so the stability, but measured by the coefficient of variation here. So basically, first relationship looking at biodiversity stability relationship. And this is from Dave Tinman, 1996. And you can see this is a negative relationship. So basically, you have to take the inverse of that to have the stability. So higher diversity, higher species richness, higher stability. Okay. So when you have a more diverse community, these communities are more stable through time. So the community biomass is more stable through time than poor communities. But this study had a lot of discussion, a lot of debate around that. And the debate basically stems from the point that actually the gradient in diversity was not manipulated, but was actually a response to a nutrient fertilization experiment. So Dave Tinman had an experiment where he had several levels of fertilization, and then with several levels of the fertilization, also a gradient in diversity. But of course, the diversity that is responding to these nutrients is one aspect, but the other aspect that is also responding to fertilization is biomass. And basically, then fertilization acts as a confounding variable because we're not manipulating diversity. So when we look at the diversity stability relationship in this experiment, basically it's confounded by nutrients. This has been a huge debate in the literature, and I think it's an important message also that I want to point out here, because there is still potentially some people doing this mistake of confounding variables within their analysis. So what it is, how it works, you have the biodiversity stability relationship here, and that's the one we are interested in. But then fertilization, in the case of Dave Tillman's experiment, was actually influencing biodiversity and the stability of biomass production at the same time, therefore acting as a confounding variable. And to make this very clear, I make this figure here where you can really see that. So for example, if fertilization reduced diversity, that's here, this red point, and reduced stability compared to the control treatments, if that does this at the same time, reduction of both biodiversity and stability at the same time, then we have this positive relationship emerging. But this positive relationship is just emerging as a response of nutrients and not this direct biodiversity influence on ecosystem functioning. And then, of course, if you have the opposite responses, an increase in diversity leads to a decrease in stability, then you have a negative relationship. So this has been a huge debate in literature, but Dave Tillman came back with this experiment, manipulating plant diversity in Cedar Creek, Minnesota, and then finding the exact same result. It's just reversed because now it's stability and not coefficient of variation. But basically came with the exact same result, but now with an experiment directly manipulating plant diversity, which is of course the only way you can get causal relationship, and that's what we got here. So this is the real first evidence that biodiversity has a positive impact on stability of biomass production. Since then, there have been many other experiments showing that. This is actually a network of biodiversity experiments called BioDepth. And you can see here, again, it's CV, so you have to think the reverse if you want to look at the stability. So higher diversity, higher stability. This is a network of biodiversity experiments, as I was saying, called BioDepth. So 8 sites in Europe were doing the exact same experiment, and then the data were pulled. But you can see that this relationship is more or less valid in all these different sites within Europe. So there is basically increasing evidence that biodiversity improve stability and basically act as an insurance against climate variability. And this is a consensus, I'm sorry. This is a consensus that has been reached, and we now have really a lot of evidence that this is happening. So the goal now is to understand how is this working? Why, when we have higher diversity, we have a more stable system? And if we know that, then obviously we can use these processes to actually, target our communities to be more stable, because we know exactly how this is working. And the processes that have been identified, I listed 3 here. That's the most common ones. But of course, there may be other processes going on. Over-yielding, species asynchrony, and species stability. And I will go through these different aspects so that you can understand how this is working. So the first aspect is over-yielding. So when you look at the definition of stability, then you will see that it's taking the mean divided by the standard deviation. So basically, if biodiversity increase the mean, or decrease the standard deviation, or does the 2 things at the same time, then basically this will lead to a higher stability. And we know also from studies that biodiversity usually leads to a higher productivity. So this over-yielding, this higher biomass production in a mixture compared to the lower monocultures or low diversity communities, this over-yielding can play an important aspect in stabilizing biomass production. And this is some evidence that this is happening. So this is the exact same figure that you've seen before. That's from the BioDepth experiment with these 8 different sites in Europe. I just switched the axis. So this is stability instead of the coefficient of variation, but that's basically the same figure that you've seen before. And now if we look at the contribution of the temporal mean and the temporal standard deviation, this is what we can see. This is the temporal mean, this straight line here, this plain line. and the dashed line is the standard deviation. And what you can see is that how this higher stability is coming, it's because the biomass first is increasing, so you have a higher mean biomass production at higher diversity. And this increase in the mean is stronger compared to the increase in standard deviation. So what I find fascinating here is that both the mean, but also the standard deviation is increasing. Both at the same time are increasing. So when you think about stability, which is the variability, the inverse of the variability, then I'm gonna say, well, is this really a true stabilizing effect? When, okay, you have higher diversity, you have higher stability, but still it's more variable compared to monocultures. So actually this stabilization factor here happened to be by this over-yielding mechanism, okay? And I find this fascinating. Why? And this is the next study. Using the nutrient network, so that's a network of fertilization experiments that are done around the world with nitrogen, phosphorus, and potassium being added in all the different combinations. And of course, we have also the control plots here. And using actually these control plots, this ambient condition, I could check whether in natural system, we have the same processes happening than in biodiversity experiments, okay? So coming back, so of course we start with the natural system, we have confounding variables, we manipulate richness so that we have the causal effect. But when we come back to this natural system, controlling for confounding variables, what do we see? And what we see is basically this figure here on the right. This is the nutrient network data, the control plot, so the ambient plot, not the fertilization. And what you can see is that we have the exact same relationship as in biodiversity experiments. And this dashed line actually here is actually reporting the results from the nutrient network. So very, very similar relationship in natural system. So what we see in biodiversity experiments, in control manipulated studies, we also see that in nature. And if we look at the contribution of over-yielding, we have quite a different picture compared to biodiversity experiments. You can see that there is no over-yielding in natural system as compared to biodiversity experiments. What we see in these biodiversity experiments in terms of over-yielding is not happening in natural system. But if you look at the standard deviation, the variability in the system, this is what I would call a real stability effect, because the variability is really reduced, okay? So we really have this decrease in variability with higher biodiversity. And that's quite a different picture compared to biodiversity experiments. And I find this fascinating, and understanding this, why this is happening, is obviously a very important component of understanding these processes. Now we have other processes that can happen. And one is species asynchrony, and I'm going to explain how this works. So I take this example again of these two species, the blue and the yellow species, and you can see through time different biomass production. It seems that these two species don't really like dry environment because they produce less biomass when it's dry, okay? And obviously, the community will respond according to the sum of these components, biomass. So if these two species vary a lot in their biomass production year after year, then obviously the community, of course, vary a lot. Now, this is for a low diversity system. We have a higher diversity system. And even if we have many more species than in this poor community, you can see that if all species basically respond in the same way, they all don't like dry environments, basically, you will see that the community are also varying a lot. Very simple. And this is called low asynchrony or high synchrony, okay? So because of these synchronous responses of these different species, then the community biomass will fluctuate a lot. Now, if we take species that respond in asynchrony, so when a year, let's say a dry year is bad for one species, is actually good for another species. This species can compensate. And if we have this compensation, these asynchronous responses can actually lead to a stabilization of the community biomass. So it's quite interesting because actually, when you look at the component species, they can vary a lot in their biomass. But because they have these asynchronous responses, then it stabilizes the community biomass. And of course, I just want to put this figure as well, and these two side by side to show actually that you don't need that many species. You can actually have a perfect asynchrony with only 2 species, and you would have a stable community. But of course, the likelihood of having this compensation, this compensatory mechanism happening, is increasing with diversity. All right. What are the proofs? What kind of evidence do we have from systems that this asynchrony, that make this process, is actually working? This is the exact same figure that you've seen before. This is the neutral network control ambient plots, and then the biodiversity experiment. And this is looking now at the species asynchrony. And you can see that in both the controlled system, the biodiversity, but also in our natural or semi-natural system of the neutral network, you can see that we have the same thing happening, which is when you have a higher diversity, a higher number of species in the system, you have higher asynchrony going on. And therefore, showing that this mechanism is valid not only in biodiversity experiments, but also in natural systems. Another process that can happen is species stability. And I was explaining before that what I find fascinating is that species can vary a lot in their responses, but if they are asynchronous, they can actually stabilize the community. But obviously, if the species are very stable, then the communities are very stable, okay? So that's just what this is showing. And of course, this can happen at low or high diversity. And there is also increasing evidence that this is an important mechanism. But overall, if we look at the strengths of evidence, and I will show it a little bit later, we have more and more evidence that asynchrony, species asynchrony, is actually usually a stronger mechanism stabilizing community compared to species stability. All right. Okay, so this is all the evidence we have accumulated from what we can call local scale relationship. So until now, what I've shown are plot scales, okay? Whether it's the biodiversity experiments or the neutral network, we have a plot 1 by 1, 2 by 2, 5 by 5, whatever the size is pretty small plot, okay? Now, the tricky thing with this is that the scale at which we are actually doing our experiments do not match the scale at which policy or management is taking place. And so this mismatch between our knowledge and the actual management makes that it's very difficult for practitioners to say, okay, now I'm going to use this result to apply this in our system because of this mismatch in the scale. So the question is, can we scale up this knowledge that we have from local scale to larger spatial scale? And basically, that's what this figure is showing. You can see this very small plot. This is, again, the Cedar Creek experiment. And can we scale it up, regional scale, but also hopefully to the global scale? So fortunately, there has been a lot of theoretical developments that allow people like me, which like to test theory, to actually assess whether this mechanism or these processes are working. And Xiaopeng Wang, together with Michel Laurent, they developed this partitioning of the stability processes, or the processes that leads to stability. And this is how it works. So remember, I showed species stability and species asynchrony. And what Xiaopeng Wang did is to take this community stability, which is also called alpha scale, this community stability, and to partition this into these two components, species stability and species asynchrony. Of course, there may be other processes going on, and actually there is some extension of this bipartite partitioning. But basically, this is saying this stability at the community scale can arise from both components, species stability and species asynchrony. And of course, given this partitioning, we can look at how diversity here at the local scale, so the community diversity, can influence these processes and then lead to higher stability of the community. So this very similar figure to what I showed before. We have one community, a green community, and then two species here. And then you can have low asynchrony, low stability of the species. This will lead to a low stability of the communities. Species stability can ensure community stability. Species asynchrony can also contribute to higher community stability. So that's the local scale, all right? And what Xiaopeng did now is to actually upscale this to this gamma stability. And he used basically the partitioning that we have from gamma diversity with alpha and beta diversity. And he applied this to stability, having gamma stability, and then the alpha and beta component. So this is the new part here, all right? And what you can see now is we have two communities, the green and the purple kind of community. And this is here, this new component is this spatial asynchrony. And what it is, you can see here that we can have these two different communities. So it's among communities, not within community, but among community. Now, if biomass production of these two communities are asynchronous, then this can also stabilize biomass production at a higher spatial scale. So that's very simple. It's exactly the same process as species asynchrony within a community, but now it's the two communities within this meta community, all right? But very similar process as the species asynchrony, just at a higher spatial scale. And then Xiaopeng Wang defined that beta diversity, so the dissimilarity in species composition among communities, can play an important role. So basically, if you have two communities with a different community composition, and if they respond differently to the environmental fluctuation, this can stabilize biomass production at larger spatial scale. So what is the evidence for that? This is, again, using the neutron network. So that's an extension, if you want, of the 2014 paper, looking at what's the evidence for these processes within natural communities. This is the alpha scale, so the community scale. And you can see that species richness plays an important role in stabilizing biomass production at the community scale, but of course also at the larger spatial scale. And this is mainly via asynchronous mechanism, okay? So that's the evidence that we have at the local scale. Now at the larger spatial scale, you can see that beta diversity, so dissimilarity in species composition among communities, plays an important role into these different responses that of biomass production of these communities to stabilize stability, to lead to a higher stability at the larger spatial scale. So what do we have? We have a local scale process here, and we have a larger spatial scale process. Landscape, regional, whatever a landscape or region is, okay? But we have these different aspects that we can then try to link to how we can use this information in practical terms. And what we learned from this local scale relationship is that if you increase biodiversity within your community, then you can have a higher stability. So how do we do that in practice? Well, intercropping would be, for example, a very good solution. You have different crops in your community, so in the field. And then if a crop does, if it's a dry environment and it's not good for a crop, and it's good for the other crop, then we can maximize community productivity, right? So that's very simple. Intercropping could be the solution. Of course, intercropping is not easy. You have to have the right machines, you have to have the right combination, so that's very difficult. I find the scaling up quite interesting here, because it's saying, well, it's not really the diversity within the plot, within your field, but it's the diversity among them. So just cultivating different crops in the landscape should act as in the same way as diversifying species within your field. Because if you diversify your landscape, then one crop can do well in one field, another can do worse in another field, but this asynchrony should lead to a higher biomass stabilization at a larger spatial scale, at the regional scale. And there are some evidence that this is happening. This is a paper from Dave Tillman with Delphine Renard, where they show this, that this is happening. Diversification of the crops in the landscape as compared to having monocultures in larger spatial scale can really help to stabilize food production. So this is an important aspect, and I think understanding these different processes, what works at the local scale, what works at the larger spatial scale, can really help us to guide advices for practitioners or managers to actually use these properties to help us to get more sustainable services from our ecosystems. So we've been from local scale to larger spatial scale. I showed you some evidence. We then did the review together with Fons van der Plas, looking at what is the strength of evidence over all studies that look at the link between biodiversity and stability and the processes undergoing this relationship from local to larger spatial scale, so that we can really assess what is the evidence for that. Do we have strong evidence that this is happening? Which processes are the strongest? And therefore, do we have a kind of consensus? So this is the review we did. And many different sites, you can see that there is a huge bias towards terrestrial plants, for example, drylands and desert and temperate grasslands compared to the other ecosystem, which obviously points also at some of the necessities to study other ecosystems and to see whether this relationship is also true in these other ecosystems. So this is a big table. So I'll try to go through. Positive effect, neutral effect, negative effect with green, yellow, and red. And you have a positive dark arrow when it's very strong evidence. Then you can have a low evidence with this kind of horizontal arrow. We don't have negative arrows, but that could happen, of course. So you can see here, I circled the strength of evidence for the contribution of alpha diversity, so local scale diversity, to species synchrony, overyielding, so the mechanism, the processes, alpha diversity to alpha stability, so just the alpha, so the diversity stability relationship in the local communities, and at larger spatial scale. And what you can see here is that for each of these relationships, we have pretty strong evidence that biodiversity overall, over all the studies that we looked at, has a very strong contribution to first stabilize biomass production at the local scale, and that species asynchrony, but also overyielding are very important processes in this system, in this process. The relationship between local scale diversity and larger spatial scale, larger scale stability is a bit less because we found only 3 studies that look at that. So we need more studies, and there are actually quite a few studies since we published this paper that are coming out. Now, if I look at the processes, you can see that species asynchrony is a very strong mechanism. So not only biodiversity promotes asynchrony, but asynchrony is one of the major mechanisms that leads to higher stability. Overyielding is also an important process, and so that's then the relationship between local scale and larger scale spatial scale, local scale and larger spatial scale stability, which is also very strong. But again, only 5 studies, but these 5 studies really agree on this strong link. Now, if we move at the larger spatial scale, you can see that beta diversity plays also a very strong role in stabilizing biomass production at larger spatial scale. And so that's the one here. And you can see that spatial asynchrony, so this asynchrony in biomass responses through time plays an important role there. And then that spatial asynchrony is linked to gamma stability. So we have mounting evidence, but you can see that some of these numbers are very low. So we tried here to make a review of what we know. And we also identified that we are really at this emerging kind of knowledge here. And again, there are more studies that have been published since we did this review. But I think we are getting into kind of some consensus, but we need more data to really make this very clear, but also to identify when it's not working, when it's working, and when it's not working, and why. Okay, so until now, I've been really just focusing on the biodiversity stability relationship. I would like quickly to also show that our global environmental changes that we are having, especially nutrients enrichment, coming back to the nutrient network data, can have strong impact on the relationship between biodiversity and ecosystem functioning, and stability in particular. So I'm going to focus on this arrow here, this one here, that basically saying some global changes that we have, I'm going to focus on nutrients, can impact the relationship between biodiversity and stability. And this is using the nutrient network data, as I was saying. And what we found is that this is the exact same relationship we've seen before, diversity-stability relationship. Now, this is under fertilized condition. And this dashed line here reports the results from the control and manipulated plot. What you can see that when you fertilize these communities, you have a decrease of the stabilizing effect of biodiversity. So biodiversity does not act anymore as stabilizing biomass production when you fertilize. And this is what you see here, this strong decrease in the positive effect of biodiversity on stability. And we have also strong evidence that this is due to this asynchrony mechanism that is changing. So this is the asynchrony, so richness asynchrony process. And this is, again, reporting this ambient condition. And you can see that when you fertilize, these asynchronous mechanisms are basically canceled out. So you sort of force the species to respond in the same way by fertilizing. So you lose this insurance. So this was the local scale. What we see at the local scale, we see this also at the larger spatial scale. So if you compare this left figure, this is the figure I showed before, scaling up from alpha to gamma scale, and this is now under fertilized condition. And if you compare the relationships from the left on the right figure, you will see that most of these relationships, most of these positive effects of biodiversity on alpha and beta and gamma stability are basically being reduced. Not only at the local scale, but also at the larger spatial scale when you look at beta diversity. So we're basically losing all this positive effect of biodiversity, whether it's at local scale or at larger spatial scale when you fertilize. And of course, understanding this is very important, understanding why this is happening, why when you fertilize, and maybe this is valid, and actually we showed in some other paper that this is valid for other global change drivers, not only for fertilization. We have this destabilizing effect. Now, future challenges. As I showed you, many ecosystems are understudied. I showed that basically terrestrial systems are the most studied system, but we need evidence in other systems. We look mainly at biomass as a process, but what about other functions? Do we have the same thing happening? Of course, mostly we look at richness. If we look at other components of diversity, we see the same relationship. When we actually pool all these different kind of processes, multifunctionality, do we see the same thing happening? Of course, linked to ecosystem services. There may be other processes. I talked about overyielding, species stability, species asynchrony. There may be other processes going on. But what we also need is actually direct experimental evidence. All the evidence that I showed you are studies that look at either biodiversity experiments or natural system undergoing some natural variation in the climate, whatever it is, temperature, precipitation, other things. What we miss is actually direct experiments manipulating both at the same time to really understand and scrutinize all these processes and understand them in details. And this is what I would like to show you with the last 3 minutes I have, which is this new experiment that we set up at Utrecht in the Netherlands, which is called BioCliVE. It stands for biodiversity and climate variability experiment. For climate is actually precipitation. We could have called it BioCliVE or something like this, but it was less cool. It didn't really work that well. So we use BioCliVE, but we focus actually on precipitation. So you can see huge experiment, 352 of these containers. So that keeps us busy. They are grouped by 4. And the reason is that we are manipulating both biodiversity, plant species richness, and future precipitation. And we do that in a split plot design. Okay. And the split plot is actually, the split plot is actually represented by these 4 different plots. These 4 different plots here that you see have the same diversity, either a monoculture or a 4, 8, or 12 species mixture. So that's our biodiversity gradient. 1, 4, 8, and 12 species. And then each of these different subplots receive a different treatment. We have a historical control, pulse press, and together, but I will show you exactly how this works. So basically, we have the historical control. That's this blue line here. So you see here, this is the precipitation for each month in the year, and it's an average over the last 60 years, I think. So that's what's raining on average. Interestingly, the more rainy months are July and August. That's what it is. So this is the historical control. So if it's raining, 60 whatever in January, this is what we had on average, all right? Now you have these red points, which are the predictions for 2085 based on the Dutch meteorological stations. And you can see that the forecast is that we have higher precipitation in winter and lower precipitation in summer. And we are applying this change. We want to understand in this future climate, what will be the impact? And this change is every year. So it's a kind of press system. Every year we have this change on average. And the other thing that we are having is that floods and droughts, so extreme events, are becoming more frequent and more intense. And this is also what we're manipulating here. And of course, we have the 2 effects happening at the same time. I want to mention that this drought that we do in the first year, it's only one time. We don't repeat it. Okay, so that's quite different from the seasonal change, which we repeat year after year. And these are really quick results. 1 last minute, I will take over time, sorry. But I really want to show you this. We actually applied these treatments only this year. We started the experiment in 2017, but to establish the communities, to make sure that they are established before doing the precipitation treatment, then it took us basically 6 years. And now we started with the experiment with treatments, but here are results for bait laminar strips. I don't know if you're familiar with that. Strips, holes, a substrate in it, put in the soil, and then you have the invertebrates that are feeding on that. And you can measure the feeding activity of this soil detritivore. That gives you an idea about the carbon cycle and how carbon is stored in the system. And what you can see, the first year after establishment, no relationship between diversity and feeding activity. And you can see that through time, these relationships are building up, and now basically it's kind of stable. I mean, stable in the sense that it stays positive. So you can see this, I find this fascinating because it's really showing when we start the experiment, it's basically soil and plants, but nothing has really happened, okay? Through time, there is a process happening, plant-soil feedback, that actually creates this positive relationship. And what I find even more fascinating, this is now the reasons for the treatments that we have. Basically, the ambient and seasonal this year were very similar. It's going to change in the future. And the drought and seasonal drought are also very similar this year. So you have these green and red lines and this purple and blue line, and you can basically sort of pull them together. And what you can see, this is the diversity, this is the feeding activity, and this is in the higher proportion of the soil. Okay, so the first I think it's like 0 to 5 centimeters, something like this. So it's the first 4 holes in these bait laminar strips. And you can see that when you have, and the ambient and seasonal are basically wet because they have a nice kind of watering system. And the drought and seasonal drought are the drought we stopped watering, okay? And what you can see is that the positive effect of diversity is vanished when you have, so you have this positive effect of diversity on feeding activity, and it disappears under drought. But what I find even more fascinating, is that when you look at this change, and this is now the lowest part, so the last 4 holes in the strips, you can see that this positive relationship that we have when there is wet condition is becoming less and less strong the deeper you go in the soil, but the reverse is happening for the drought treatment. So it seems like these communities that are working well in wet environment when everything is great, they are working on the topsoil, and this shift to the lower part of the soil when it's drought, which makes sense. But what I find fascinating is that basically this switch in the level actually stabilizes the whole system. So there's a kind of insurance by shifting the depth at which these microbiome communities are working, or the invertebrate activities are working. This shift actually ensures that there is overall a positive relationship. So I find this fascinating. This is really just new results. I still have to understand why this is happening, etc. But I wanted to give you a quick glance on some results. And I'm over time, sorry for that. These are the projects that are ongoing. Many people working on this BioCliVE experiments. So I want to thank them also for that. And I would like to thank you for your attention, and I'm happy to take any question.
Wow, thanks a lot for this super nice, interesting talk, Yann.
Sorry, I went over time.
5 minutes, 4 minutes. I think that's okay.
I started later also.
I think you did perfectly. Are there questions? We also have people on Zoom, so people on Zoom can type their questions in the chat, and Nadia will let me know. But maybe first we start here in the audience. If no one has one, I can also start, and you can think during this. I was wondering, we always think that stability is something good, so we want high stability. Yep. But if, for example, you have 2 communities, they are both doing pretty well when it's nice and wet, and if it's dry, then maybe one community drops. Or let's say they all are the same when it's dry, but when it's good conditions, one community has a higher biomass, which is actually good, but it would have a lower stability.
Yeah.
Should we then rethink stability in some cases and only consider those losses, or when it's lower than average? Yeah,
well, that's a very good point. I mean, there is a kind of difference in when you look at the longer term stability, which is this temporal variability invariability that I've been focusing on here, and this shorter time aspects. And actually, this is also something that I find fascinating, because we all have kind of different understanding of what is stability. And actually, when I present my results, there is a lot of people, or especially in the media, they are always referring to resilience. And I think people understand stability as being, let's say, maybe more like the general audience understand stability better as in terms of resilience, which is for me just one aspect. And I think if we look at the literature, especially the ecological literature, because there are different definitions depending on where you go, in which field you go. But in ecology, when you look at resilience, it's a very specific aspect of stability. And it's really the ability of a function or whatever process in the system to bounce back to the original state. It's just one aspect of stability. And this, of course, contributes to the temporal. So this, and of course, the resistance is a very important aspect, how this ecosystem resists against the change, how it bounces back. And these 2 things cumulated basically make the stability in general. But what I find really interesting here in this debate is that first, of course, there is this definition of it, what we call stability. Is it resistance or the long term? But also, coming back to the original state is great, but it's not just how you come back to it, it's how you went there. And this drop that you see is the one that contributes to the temporal one, these changes. So resilience, if you say a system is resilient, you're going to say it comes back to the original state. Great, you're happy, it's stable. Well, it depends. If you have a low resistance, so a huge drop, and it comes back to original state, it's going to be less stable than if you have a high resistance for the same resilience. So resilience is not just it. There is really the pathway through which it gets there. And I think this is something that I've been learning more and more, is that there is this disconnection in our definition and how also people really get to this point. And maybe one day I'll publish a paper explaining this or trying to disentangle what is the contribution. And I think there are other people working on this. But I also see in some of the papers that we publish, there is also a mismatch sometimes even in this definition. It's very difficult to keep it really strict, or sometimes you will call it resistance and then it's actually recovering. But you explain it's recovery, but it's still called resilience. And that sort of makes, that adds up. When I was talking about the definition of stability, that sort of adds up into the mess of what is stability, and therefore also how people understand it. And that makes it a bit, I think, tricky for us to really get a clear message.
Cool, thanks. Maybe now there are some questions in the audience, Steffen.
Thanks for the nice talk. I had one question related to the land use intensity factors, because from the German biodiversity exploratories, we know exactly what you showed, that community stability is more related to asynchrony than to species richness, but also that land use intensity crushes these relationships.
Yep.
And have you already thought of, instead of only manipulating the N factor, also the other, to manipulate also the other factors like mowing intensity or grazing intensity?
Yeah. So obviously I thought about that. The question is the money, time, etc. into doing that. But it's true. So what I showed for nutrients, the question is it's valid for other global environmental change that we see. The study that I can refer to that I've been working on was a study where I looked at different experiments manipulating different aspects of global change: nutrients, fire, herbivory, water. I think that was it. At the Creek in Minnesota. The advantage of this is that all these experiments are run at the same place. And so climate, all these different types of soil, etc., are more or less controlled for. So it's kind of, controlled. And the advantage of this is that they also have two biodiversity experiments, the Big Bio and BioCon. And so we can actually compare what we see in these biodiversity experiments compared to what we see in these experimentally manipulated experiments. So what we see is that the relationship that we have between biodiversity and stability in this controlled experiment, we see this also happening in these different experiments manipulating different global change drivers. So what we found is that when a global change drivers decrease, a global change driver decrease diversity, it leads to also a decrease in the stability. Whether it's nutrients, whether it's fire, whether it's herbivory, whether it's water, we see the same thing happening. What I find also fascinating there is that maybe I do have the, actually this is the slide. Oh, lucky me. So this is actually the slide showing it. So you can see we have different treatment, actually, of course, also carbon dioxide manipulation. So all these different drivers that have manipulated there. And these are the two biodiversity experiments, BioCon and Big Bio. And this is the relationship that we have in these two experiments, the controlled experiment. And this is what we have in this natural system. So in this controlled experiment, a decrease in diversity leads to a decrease in stability. Now, in this experiment where we manipulate these global change drivers, and I looked at temporal change, if diversity is reduced through time, stability is also reduced. And what I also find interesting is first there is a kind of like, the red points on the rest. And there's always the critics that various experiments, have like huge changes in diversity. And so it's unrealistic. Yeah, it's true. You can see that here. That's right. But this relationship is sort of going, in the same direction. So even if it's indeed unrealistic, it's just a higher level that goes in the range of what we see actually in natural systems. So I find this also quite interesting in the whole thing. So it shows that it's sort of general because we see this among all these different global change drivers. And it's also quite similar to what we see in various experiments in this small control system. I don't know if it answers the question, but yeah.
Great. There are questions on Zoom. Maybe, should I read them? Or do you want this one? Okay, so how do I convince my neighbor who is a conservative farmer? If he fertilizes his grassland, he would harvest more anyway, even if he loses a few species.
Yeah, that's the core of the whole thing. And that's a very good question. I'm happy whoever asked this question, because it's an interesting thing. So obviously I talked about the over-yielding. There was actually a very nice paper written by Carnus. I don't know his first name, but together with John Connolly. And I think this is a good reference. Why? Because they really debate whether this invariability or stability, as we measure it, like mean to standard deviation, is it the right measurement? Because of this debate of whether it's matching what managers or farmers like. So of course, for a farmer, if you fertilize, what's happening is you basically increase so much the over-yielding, right? You just increase biomass production. And this increase in biomass production also increases the variability of it. It goes together. But the benefit of fertilizing on the mean, on the over-yielding, is so high that basically, even if you lose species, even if you have high variability in biomass production, it's still better. It's still more stable. And that creates a kind of mismatch between what I show you here and what farmers experience in the field, which is also something that is a very important message. And I'm very happy that this is discussed because, yeah, what is best? And it's true for a farmer, fertilizing, because you increase so much biomass, you basically have your stability happening. But I think it's missing the fact that we just having, if you look at the multifunctional landscape, where all the different functions are more or less at the same level, then what we do is we squeeze this whole thing. So you reduce all the, for example, you have increased nutrient leaching, you decrease maybe the health of the cows, all these kind of aspects that are there, you decrease them to maximize one function. And I think, as if we think about nature and how it works, personally, I don't think that's the way to go, and I understand the farmers and why they do that. But I think there are other ways to do it. And what I'm showing here is that biodiversity is key towards that. Of course, we have to manage system with the right species. And this is what I'm trying to understand is which species are working. There is very nice work from like Matt, I think it's Andreas Lüscher from the Agroscope, for example, that look at this, what is the best combination of species, so diversity, increase in diversity in these agricultural fields, pastures, for example, to really maximize biomass production and their stability. And what they show actually is that when you can actually reach the same level of biomass production with high diversity. It's 4 species versus monoculture, so it's not super high, but still it's better than 1. You can have the same biomass production with 4 species compared to fertilized communities with, I think it was 45 kilogram per hectare or something like this. I cannot remember the exact numbers, but you can basically have the same effect. So biomass, having a higher biomass production and more stable, but with diversity as compared to high fertilization. So there are solutions, I think, to basically decrease our impact on ecosystem, to benefit all the other functions that we have, and to also have some positive effect on biomass production. But I think maybe one last thing is that it's a trade-off. Huh? So when I also say this, I realize that for farmers, there is a cost. Even if you use diversity wisely, I don't think you will ever reach this kind of high biomass production that you have in this intensive system. I don't think it's possible. But I think we should also think about the way we manage our ecosystem to give back to nature and maybe have subventions from governments, etc., to minimize our impact. So there is a very difficult kind of trade-off there.
Thanks. Are there more questions from the audience? Noemi.
Thank you. I have a question. Did you have the occasion to analyze data from experiments like BioDevs, for instance, or more of those? Are those experiments long-term enough to test if stability decreases or increases with time, as biodiversity effects are usually increasing with time, also in manipulative experiments?
Yeah, that's a good point. I think there was a recent study from the Yann experiment from Cameron Wag, I think showing that stability increased through time, if I remember correctly. I would have to go back to that, but maybe I don't have it here. Yeah, I don't have it here. I don't think I have the figure. No, this one almost. No. Okay. But in the 2020 paper looking at neural network data, we can see that stability through time is sort of getting steeper. So the diversity stability relationship through time is sort of getting steeper. When I tested for interaction, I didn't find one. So maybe it's not long-term enough to really get at this. But my impression, and also based, I think, again, we'll have to double check this paper, but I think Cameron Wag from the Yann experiment, they show that stability is increasing through time. So based on this, I would say yes, but double check what I'm saying. And also longer term, indeed, we need longer term data, and that's very important. Yeah, these longer term experiments need to be available to test this. Yeah.
Maybe we can have one last question by Kurt.